Today we're going to be covering concept 3.4, regulation of metabolism occurs by regulation of enzyme. Textbook pages 53 through 57 from Principles of Life, second edition for the AP course by Hillis et al. Now, it's important to remember that enzyme catalyzed reactions do operate in metabolic pathways. These are very, very complex. These are not one step and it's done. Cells have hundreds of enzymes that participate in interconnecting metabolic pathways, forming a metabolic system. Now, figure 3-17 on page 53 in your text gives a metabolic system that we see here. Okay, so each one of these dots right here represents a different reaction, and you can see how they are all connected. So we have nucleotide metabolism, metabolism of cofactors and vitamins, amino acids, other substances, ATP production, all of that. Okay, each line represents a catalyzed metabolic reaction. And you can see that a lot of them come together in one place, like right here, the metabolites and reaction pathways overlap and intersect. It's very, very, very complicated. Okay, so complicated that it's very difficult for us to actually understand, all of them independently. There are some people who focus exclusively on carbohydrate metabolism in their research, others that do, lipid metabolism. All of that, but you can see it's very, very, very complicated, very, very branched. So systems biology is a new field that describes all of that, and because of its complexity and because of the sheer quantity of reactions, we actually see that typically computers are used to study all of this because of the high levels of complexity. So cells can regulate metabolism by controlling the amount of an enzyme, and they often do this with the ability to turn synthesis of the enzymes off or on. The activity of enzymes can be regulated as well. Obviously, it's going to be a little bit quicker than trying to turn it off or turn it on. So we're going to talk about different ways that enzymes are inhibited. So chemical inhibitors can bind to an enzyme and slow reaction rates. Natural ones regulate metabolism. Artificial ones are used to treat disease, kill pests, and also study enzyme function itself. So let's start with irreversible inhibition. So this is when an inhibitor will covalently bond to the side chain in an active site. It's permanently inactivated. Insecticides work this way. They will move into an insect and they will cause changes in their digestive pathways or metabolic pathways, similarly to what we saw in that metabolic systems map a minute ago in figure 317. So in this case, figure 318 on page 54 of your text, DIPF, this is an inhibitor, will come in, reacts with the hydroxyl group here on a serine in the active site of the enzyme. This covalent bonding will result in an irreversible inhibition of the enzyme, meaning this cannot be reversed. So this is permanent change of acetylcholinesterase. So reversible inhibition. So sometimes we do want to, obviously be able to reverse this reaction. We can have a competitive inhibitor, which will come in, binds to the active site, no reaction occurs. It'll compete with the natural substrate. We have a non-competitive inhibitor. This will bind at a site other from the active site, causing change. It's going to prevent substrate binding or slows the reaction rate. It's going to change the shape of the active site slightly. Okay, so we have a competitive inhibitor here. We'll bind at the active site. We have a non-competitive inhibitor here. You can see it closes the active site, preventing the reaction from occurring. So sometimes we have allosteric regulation. So this is when a non-substrate molecule binds to a site other than the active site, changes the enzyme shape will change and alters the chemical attraction for the active site. So this can either activate or inactivate enzymes. Okay, so this can be done by non-covalent bonding, this reversible, or covalent bonding. We can have things like phosphorylation. We can add a chemical group. Again, these are all reversible. So figure 320 on page 55 in your text shows this. So we have this activator come in. When the activator comes in, it will open the active site, so to say. The substrate will come in, it will bind, and we get our products. Okay, in this case, in figure 320b, we have phosphorylation. So in this case, we have this phosphate group come in, it binds, opens the active site, again, substrate binds, and we produce our products. So phosphorylation is an important regulatory mechanism, and it can change a hydrophobic region to hydrophilic. Enzyme will twist, expose the active site, has several changes that can occur. Protein phosphatases can reverse the process by removing those phosphate groups, therefore inactivating the enzyme again. So this protein phosphatase will come in, it kicks this phosphate group off and inactivates the enzyme. So metabolic pathways, the first reaction is the commitment step, so to say. So we can have feedback inhibition, meaning end product inhibition. This is incredibly common in metabolic pathways. The final product itself acts as an inhibitor to the first enzyme. We can see this in figure 321 on page 56 in your text. So the first reaction is the commitment step. So in this case, we have threonine. We are trying to get to isoleucine over here. So we have this commitment step comes in. Each of the reactions, it's catalyzed by a different enzyme. Each forms a different intermediate product. Once we reach our end product right here, when we reach a certain amount, that itself will actually regulate this reaction because, again, these are energy expensive reactions. If we don't need to produce more isoleucine, once it's sensed that we have enough, we will turn this reaction off. This pathway stops so that we won't continue producing them. Okay, so pH can affect them. Okay, so acidic side chains that generate hydrogen ions can become anions. Basic side chains will attract these hydrogen ions and become cations. Okay, so pH will affect a lot of these as well. One of the primary ones that we get into that we know about is going to be cellular respiration and the production of lactic acid. So in this case, we have glutamic acid plus a carboxyl group. We will split this carboxyl group and it will enforce the law of mass action. So the higher the hydrogen ion concentration, the more the reaction is driven to the left. Okay, this way, you remember these arrows mean that the reaction can go both ways. And the more the reaction is driven to the left, the less hydrophilic form. This can affect enzyme shape and function, again, due to the concentration of hydrogen ions, or commonly whether it is acidic or basic. So protein tertiary structure as well is very sensitive to the concentration of hydrogen ions in the environment. All enzymes have an optimal pH reactivity. Okay, so when we get into situations where it is too acidic or too basic, we start seeing that tertiary structure of proteins changing. And again, that's the third level where we start having several individual proteins come together. Temperature affects this as well. Warming increases rates of chemical reactions, but if it gets too high, non-covalent bonds begin to break and activating enzymes. We can see this as well. If we get too cold, we can't continue either. All enzymes, again, just as with pH, they have an optimal temperature for activity. Okay, so we can see this in figure 3.22 on page 57 in your text. With our reaction rate, pepsin in this case is going to be used in the stomach. It's to help break down proteins, and it operates in a very acidic very acidic state. If the stomach acid becomes too basic, starts moving towards more neutral, pepsin doesn't really function, but at that point we can have chymotrypsin and arginase come in. They will do the same job. And right here we have the generalized model for reaction rate reaching a maximum rate of efficiency based upon temperature. So isoenzymes can catalyze the same reaction but have different composition and physical properties. They may have different situations for different pHs, temperatures, whatever the case is, allowing an organism to adapt to changes to its environment. Without these isoenzymes, we would be stuck in this single situation where we're only good at this pH, but what if we have a situation where we move beyond that? Let's say stomach pH is around 4.5 for whatever reason. We still need pepsin to function, but an isoenzyme can come in and help allow for better adaptation to that. If you have any questions, make sure you put them in the comments below or bring them to class.